just want to catch your point about being interactive. Of course, we cannot address any question at this moment, but all the attendees have the opportunity to write their question directly in the Q&A box. I just remind you to write the question with your name and surname and with the name of the speaker. We will choose the most relevant question at the end of the session, and we will try to address all the questions later on these days. Okay, so now it's time to move to the next speaker, Yann Devos from the European Food Safety Authority. Yann is a senior officer in EFSA. He's a strong expertise in environmental risk assessment of genetically modified organisms. Today, he will try to give you some ideas, fundamental ideas about environmental risk assessment of regulated products under the EFSA remit, current challenge and future direction taken by the European Food Safety Authority. So Yann, the floor is yours.
So many thanks for having us presenting and giving you a flavor about environmental risk assessment. So we are very honored to be part of this summer school session, and also very grateful to kick off the discussion and session about the environmental safety and biodiversity. So we also wish to thank all those who worked behind the screens. So they did a wonderful job in organizing, shaping the event. So congratulations to them already in advance. And also welcome in my living room. So that's the place I'm standing in. So welcome on board. Okay, let's get moving. So what we are going to do with this first presentation is to give you a flavor of what EFSA is doing in the field of environmental risk assessment. Then we are going to use genetically modified organisms as a case study, just to illustrate some of the concepts that are linked to environmental risk assessment, but also some of the risk concerns that we address. And then we will finish with a wrap-up where we tend to reflect on the current challenges, but also reflect on the future directions that EFSA is going to take in the field of environmental risk assessment. And this presentation should be seen in complement with the presentations of Domenica and Agnes. So they will talk about pesticides, pests, a more holistic approach to environmental risk assessment. So the three talks should be seen as a whole and complement each other. So keep that in mind. So let's start with EFSA. It has been mentioned before. So EFSA is the reference body for risk assessment of food and feed in the European Union. And we are supposed to provide independent scientific advice to the risk managers and also the policymakers on food and feed safety. We also provide timely and independent risk communication. And as mentioned before by Bernard, we also promote scientific cooperation because working together is key to exchange expertise, data, and address bigger challenges together. In terms of the breadth of activities that we cover the entire food chain. So it goes from the farm, the field, to fork. And in that food chain, you can encounter a set of stressors, but also regulated products. And some of these are subject to an environmental risk assessment. So you might find it a bit strange that a food safety authority is also dealing with environmental risk assessment related issue, but it's the case for feed additives, genetically modified organisms, but also plant protection products and the quarantine plant test. So for these type of regulated products or stressors, an environmental risk assessment is being performed. And what does that entail? Well, the idea is to investigate whether those activities are having an adverse effect on biodiversity, ecosystems, or also ecosystem services. And if that's the case, it's being considered and mitigation measures are being proposed to the decision makers. And in type of the activities that we consider, so we have 5 different categories. The first one really relates to the assessment of pre-market registration applications, but also renewal applications if something was already on the market and needs to be renewed. And the idea there is that you submit a dossier to obtain an authorization to put something on the market. And in that context, an environmental risk assessment needs to be performed, where in that context also EFSA plays an important role. The other big category relates to the impact of quarantine plant pests. So also here EFSA is sometimes being demanded by the European Commission to assess those impacts and also to assess the environmental impacts of the associated risk management options that might be proposed to limit the spread of these quarantine plant pests. The third category relates to the assessment of post-market environmental monitoring activities. And this means that you have put something on the market, so you have the authorization for that, but you need to continue monitoring the product that you put on the market and investigate whether it's having an adverse effect on the environment or not. That's the case for GMOs in Europe. And the fourth category relates to the development, but also the revision and the update of risk assessment guidelines, which can also contain or entail cross-cutting guidelines. And the last category relates to the development of risk assessment tools to advance risk assessment, but also to gather data to feed those risk assessments. Now we move to the case study of GMOs, just to present you some aspects and concepts related to environmental risk assessment. So for those who are not familiar with the GMO, well, it's an organism that has been genetically modified in such a way that it's not possible naturally through crossbreeding or natural recombination. And these products or organisms are subject to regulatory oversight, meaning that you cannot use them without prior authorization. So you need to have an approval first to be able to commercialize those organisms or the derived products. And in that process, EFSA is responsible to perform the prospective or the pre-market risk assessment. And that assessment is really designed and tailored to protect human health, animal health, and the environment. And there is a very complex legal frame in place, which is addressing different type of uses, different aspects of your GMO. So it goes from contained use to labeling, traceability. But today's presentation is going to focus on the deliberate release of a GMO into the environment. So it's the intention to release it in the environment. And also, for instance, if you grow a GM plant, the intention is to use it as food or feed typically. So that is also one of the intended uses that is being covered by the GMO legislation. And do not believe that there is only one GMO. The reality is that you have a huge diversity of GMOs and they differ in different dimensions. So in terms of organisms, we talk about microorganisms, plants, animals. Within the animals, you have fish, birds, mammals, insects. Then you have the methods that are being used to obtain the GMOs that are also evolving regularly. So we have the traditional methods, but also genome editing is now entering the picture, and you might have heard about it in the news recently. And also the intended traits that are being conferred to the plants or the GMOs are very diverse. So it can go from resistance to pest diseases, so that the plants, for instance, are less subject to those diseases or herbivory. If you have a herbivore that is eating the plant, you have also tolerance to abiotic stress, so drought to salt, so that you can grow plants in different conditions. Increased yield potential is also one of the intended traits. You can also change the composition, enrich the crops with vitamin content, etc., etc. The modes of action of these traits is also very diverse. So we can silence things through RNA interference or through genome editing. You can also play with the regulation of genes through transcription factors. And then the final aspect or dimension that is relevant is the intended use. So we mentioned it before. So it's food, feed, but also the deliberate release into the environment. And when you talk about deliberate release into the environment, it could be the cultivation of a crop, but it's also the import of materials from overseas where they might be growing the crops, and seeds could be imported to Europe, and that material could then end up as feed for the animals. And it's the combination of those different dimensions that you need to consider in your risk assessment. And it's clear that a single GMO doesn't exist. So you have a bulk of GMOs that are all specific. So when you perform your risk assessment, you will have to account for that diversity on a case-by-case basis. And in terms of the aspects of the risk assessment that we considered, they are very broad. So typically, we start with a stepwise approach. So in the first step, we look at the molecular aspects of the GMO, try to characterize things to identify hazards. Then we move to a type of compositional assessment, but also looking at the phenotype and the agronomic performance of your crop, if you talk about a crop. That material and information is then fed into a food and feed safety assessment, where we consider allergenicity, toxicology, and nutritional aspects. And the final part is the actual environmental risk assessment, and this is the focus of today's presentation. In the following slide, I will give you a hint about the type of risk concerns that we consider in our environmental risk assessment. So the first one relates to the persistence and the invasiveness of the GMO that you deliberately release into the environment. So the concern in brief is that your plant, for instance, would become more weedy. So it's more difficult to control. It might reappear in subsequent years, or it might also go wild and move outside your cropped area and perhaps invade natural habitats. So these are the potential scenarios that we consider in the risk assessments that we perform. So what we look at are all the limiting factors that are limiting the spread and the invasiveness of a GMO, and then we tend to see whether the intended trait might relieve or release the GMO from those limiting factors. And typically, we observe that one intended trait is not enough to ecologically release a GMO from all the limiting factors that drive the behavior of the GMO. The next one relates to vertical gene flow, and the concern here is that the transgenes would be passed on to cross-compatible organisms, which can be weedy or wild relatives. And the concern is not necessarily the transfer of the transgene, but is that the transgene, once acquired by the recipients, would give the recipients an increased fitness or a reduced fitness. If it's an increased fitness in a weedy relative, then you might exacerbate a weed problem. And if you reduce the fitness of a wild relative that you consider valuable, it might be problematic because you might want to protect that species and you might not like it to be reduced in number. So this is addressed under the vertical gene flow angle. And the same applies to the horizontal gene transfer. But the difference here is that we are no longer talking about sexual transfer, but it's really the transfer from a plant to microorganisms. And the concern there is that once the microorganisms have taken up the transgene, they might spread it very quickly. And that could occur in the soil, so in the rhizosphere of your plants, but also if you consume that material, there is potentially a possibility for transfer in the gut microbiome of the animals that consume the material. These are also aspects that we consider. The overall message is that the transfer is most unlikely, and nevertheless, we look at possible consequences of that transfer in our risk assessment. So we typically consider some worst case assumptions when we perform the risk assessment. The other aspect relates to target organisms. So these are pests that are affecting the yield. So you might want to get rid of those. So you make the crop, for instance, resistant to those pests. But in the long run, you know how evolution goes. I mean, the pests evolve resistance and you lose the benefit of the GMO. So this is a concern that can happen in the long run, especially if you grow the same crop over and over again without rotating it. So you really select for resistance to evolve. So this is being considered. Similarly, we consider impacts on non-target organisms. So these are the organisms that you value. So it can be butterflies, beetles, and they are valued because we find them pretty or because they have very important functions to play in your ecosystem. And therefore, we do not want them to be harmed. And so this is also being looked at. There are different routes of exposure. So if you see a plant, the transgenes can be expressed in different tissues depending on the crop you consider or the event you're considering. And different insects, arthropods can feed on those different plant parts. So you see herbivores eating on the plant, so they can be exposed to the toxin that is being expressed by the GMO. And then these herbivores can also pass on, in some cases, the toxin to higher, insects that occur in higher levels in the trophic chain, because they are predating or they are using the herbivores as a host. And that type of scenario of indirect uptake is also being considered in the risk assessments that we perform. Yeah, another one relates to human and animal health. So the focus here is the accidental intake of some material. So if you have a carnation, you might have some petals that might end up on your plate by accident. So is that a problem if you consume it? And then also workers that are handling the seeds that come in could be exposed and inhale, for instance, some dust from GMOs, or also come into contact when they run into the fields to spray, for instance. So these aspects are being considered as well. Now we have the soil. So keep in mind that the soil is just more than a black box. So it's also a very complex system. And we look at impacts on the biochemical processes and also the abiotic environment. Farm management practices is also being considered. So you could imagine that if you grow a crop, you can change some of the farm management practices that are linked to the cultivation of that crop. So this could have an impact on your biodiversity and the environment, and this is also being considered. Initially, there was a gray zone on whether GMO risk assessment should consider the pesticide impact, but this has now been clarified. So pesticide usage is now moved on to the pesticide legislation. So we are no longer considering this in the risk assessments that we perform. An important aspect of the risk assessment is the problem formulation. So this is the first key step that you go through when you perform an environmental risk assessment. And the problem formulation is designed to frame the whole exercise. And it will help you to focus your environmental risk assessment on those aspects that are relevant for regulatory decision making. And the context is being provided by policy. So it's the legislative framework in which we operate. And how does it work, the problem formulation? So you have heard all the type of risk concerns that we consider when we perform risk assessments. But the question is, concretely, how do you do it? Well, in the GMO area, applicants are asked to perform these kind of problem formulations, and they follow more or less 5 steps to achieve that goal. And the first thing they do is to identify the protection goals that are outlined in the legal framework, and they use that as a starting point to understand what it is that we need to protect and that doesn't need to be harmed. And then the next challenge is to make that operational, because some of these protection goals are very vague and broad. So we talk about biodiversity, this is a massive concept, or we talk about ecosystems. So you need to translate that into something that is operational. The next step is to devise plausible pathways to harm. So what you do is to have an event, cultivation of a GMO, connected to the type of harms that you might not find acceptable, and see whether your GMO is able to lead to that harm. Then you formulate risk hypotheses, you identify the information you need to test those, and then in case you miss information, you develop an analysis plan to figure out how you can generate the missing data. So in the next slides, we will go step by step through these different categories of actions. So the first action is to identify the protection goals. And typically we talk about human health, animal health, environmental health, soil health, water quality, but it entails biodiversity, ecosystem services, but it's too broad. So you need to be more specific and need to translate them into something that is operational for use in your environmental risk assessment. And typically we try to identify the intent ecological entity attribute we want to protect. We also reflect on the maximum tolerable impact, and then also try to reflect on the spatial and the temporal scale of protection that we strive to. And EFSA has published an opinion on this, where we have a stepwise approach to guide applicants through this process. So once you have defined what it is that should be considered harm, you can start devise your plausible pathways to harm through a chain of causal events. So event A is the cultivation of your GMO. Then you have a number of steps that need to take place for harm to occur. And the next step is to formulate the risk hypothesis. So the question is it possible to move from event A to event B? If that's the case, you want to identify the type of information that enables you to check whether it's possible to go from event A to event B. And if you miss the data, you need to figure out how to generate those data. And it's important to know that the testing of these type of risk hypotheses can be done with this existing information. So it doesn't mean you have to reinvent the wheel each time. So there is a lot of information available in the scientific literature that can feed that process. So what I just presented might be a bit abstract. So I've tried to present a case, an example of how we can implement this in practice. So what you see on the left side is a maize plant. So assume that this is a maize field and that this maize is a BT maize. So it's expressing a toxin that is supposed to kill a target organisms, which can be a comb borer that is eating on the plant and reducing the yield and having that type of adverse effects on the plant. And then what we want to avoid is that we are harming valued butterflies. And the reason for that is that the comb borers are butterflies and that the toxins that are expressed in the GMO are not necessarily specific for the comb borers alone, but they are specific to all Lepidoptera, so to all butterflies and moths, meaning that all butterflies and moths that would be exposed to that toxin at sufficient dose might suffer harm from it. And that's what you see on the right side. So you see a larvae of a very valued species that might be harmfully impacted by the toxin. The question is how? How do you get from the maize plant to the larvae of a valued butterfly that might not occur in the maize field itself? So that's what you try to map with the pathway to harm. So what you understand is that you have a BT plant. The BT plant is expressing the toxin in different plant parts, including the pollen of maize. And the pollen of maize can be spread with wind and so shed into the environment. And when the wind is spreading the pollen, it can fly away and fall in the maize field, but also beyond the boundaries of your maize field. And there it can drop on plants that are being used as a host plant by your valued butterfly. So imagine that you have the larvae of that butterfly feeding on the leaves of the host plant, and those leaves are being dusted with the pollen that was brought by the wind. They might get some level of exposure to the pollen, which also contains a toxin, and thus be exposed to the toxin in the pollen. So that's your pathway to harm. And for each of these different steps in your pathway, you can design a risk hypothesis. So we go from your BT maize plant to BT protein produced in the pollen. Then the pollen is being dispersed to the neighboring fields and might drop on leaves of your valued butterflies. Then you see that the larvae of that butterfly are eating those leaves and thus the pollen that is deposited on it. And the BT protein expressed in the pollen might be toxic to the butterfly if consumed in sufficient amount. And that could harm your butterfly, so you have a reduced number of larvae observed, which is not acceptable. So how do you test for that? So the first risk hypothesis you can put forward is to indeed check whether the pollen is expressing any toxin. So you can check that and have a pollen expression study to confirm whether you observe any toxin being present in the pollen or not. If you observe that there is some toxin present in the pollen, then you move to the next step and you can assess the next step. So the question is, do you find some pollen being present on the leaves of the host plant? You can test for that. If that's the case, you continue the pathway, and etc., etc. So throughout that exercise, you try to really streamline your process. And then you follow a tiered approach. So when you need to generate data, you're going to start from a worst case scenario where you expose the valued species to a high level of the toxin. And then if needed, you refine the exposure to move towards more realistic exposure. And the reality is that, yeah, testing it on the controlled condition reduces the random effects and the noise you might observe in field. So that's a bit the trade-off. But so throughout the process, you move from worst case conditions to something that is more realistic if needed. If on the worst case condition, you don't see any adverse effect, then no further testing is needed because you assume that on the then it would be safe for lower levels of exposure. Domenica is going to tell you more about this. She has a wonderful slide to present this. Then also, once you have done your risk assessment, you might have identified risks and perhaps uncertainties that remain. So those need to be addressed. And in the GMO area, we address them through risk mitigation measures. So we propose options on how you can reduce identified risks. And if uncertainty remains, there are also means to handle that. And monitoring is one of those goals. So if you allow for the crop to be grown, then you would have to follow it up annually to see whether you see any unintended effects occurring and whether the assumptions you made in your environmental risk assessment are still valid. Important point is that environmental risk assessment is not ecological research. And the reason for that is that the environmental risk assessment and ecological research differ in purpose. So the purpose of an environmental risk assessment is to reduce policy uncertainty. And I think your ecological research might be deemed to reduce science uncertainty. And so it's important to realize that your environmental risk assessment is a policy-led activity that is supposed to support regulatory decision making. So it's not an activity that is curiosity driven. So it's not supposed to maximize the production of knowledge for the sake of it. So you're really designing the whole approach to deliver data that helps the decision makers to come to a conclusion. And also the sources of the problems that you address are different between the two fields. So you might be testing for no differences, but I think in an environmental risk assessment, you would be assessing and testing for unacceptable risk. So that would be the position you take because you are driven by a policy context. And then also the methods used to testing some of these hypotheses might be different. So I think in a risk assessment approach, the tendency is to go for a more targeted approach and to use indicators of unacceptable risks. And these untargeted profiling approaches might be more designed to look for no difference effects, and this might complicate things. Current challenges and future directions taken at EFSA. So, I mean, you have a number of drivers of change captured here in the slide. Bernard already mentioned some of those. And the idea is that there are some triggers to justify the need for change when we talk about environmental risk assessment. And that change is supposed to move towards a more holistic, integrated approach. I'm running out of time, so I'm not going to single out each one of them, but there are scientific reasons, also societal reasons, and also policy reasons to perhaps rethink some of the approaches that we have put in place to perform environmental risk assessment and perhaps add another layer to the whole concept. And this thinking is taking place at EFSA. So we are having a reflection on how to frame and shape the future strategic goals of EFSA, and also specific considerations have been provided on the future of environmental risk assessment in that context. And what's the goal? Well, the goal is to move from the current situation and to transition to a more integrated, holistic approach for environmental risk assessment so that we get prepared for the new generation of environmental risk assessment. And the idea behind is to be more realistic in the type of assessments that we perform. So we would like to better consider landscape aspects and also population level related aspects so that we connect better realistic scenarios and integrate them into the assessments that we perform. We also would like to acknowledge that the single substance and use assessments have their limitations, because in reality, there might be an exposure to multiple regulated products. So in reality, you know that pesticides might be applied at different moments in the field, they might be combined. And so there is a need to perhaps consider that in a more integrated aspect, so that we also consider cumulative effects in the assessments that we perform. And on top of that, you have environmental stressors that are pushing the system to its boundaries. So we have climate change, these type of aspects also would have to be integrated in one way or another. The third element relates to the feedback loop. So we have to put more emphasis on the monitoring data, the vigilance data, the surveillance data, because they capture what's happening outside and might help us to use that information to transition to something that is more realistic. And that cannot be done alone. So we need to develop new tools. We need to have access to more data. We need to share expertise, and therefore there is a need to foster cooperation and work more with others. And that's why we are thinking to establish a partnership on this aspect. And hopefully this will help us to bridge the regulatory silos that we encounter in the field of environmental risk assessment. First steps have been taken at EFSA level. So we have a roadmap that is being under development, and it's the University of Parma that are taking the lead in developing the roadmap, and that roadmap will guide us and give us some indications on how to prioritize the next steps. Other themes that are being put forward and that will be addressed by Domenica and Agnes are the bees. So keep that in mind. Just acknowledge the contributions of all the colleagues involved in the process. So you have the names, you can read it. And then also, if you are interested in the topic of more integrated systems-based environmental risk assessment, then make sure you connect to the One Health conference that we are organizing. It will take place during the 21st and the 24th of June, 2022. You can connect online, but also participate in presence in Brussels. So keep that in mind, note it in your agenda. And then just to let you know that the B work that has been accomplished so far and that is being pursued is paving the way of how EFSA is intending to move environmental risk assessment forward. And both Domenica and Agnes will talk more about this in their presentations. So thank you very much for your attention. So the floor is to you, Chiara. Thank you.
Thank you, Yann. Thank you very much for this comprehensive overview of the topic of today.